Hello, hello, everyone, and welcome to React 19 Virtual Rounds. I'm Angel Lopez, the Provider Network Officer here in New Jersey. Don't hold it against me. I just am thrilled to have a very special guest here this evening with us, who agreed at the last moment to do that. So I'm truly grateful. Thank you, Asher, for doing this at such short notice. So React 19 is a science-based nonprofit organization offering financial, physical, and emotional support for those suffering from long-term COVID-19 vaccine adverse events globally. Our mission is to bring healing to the moms, dads, friends, and loved ones who are facing life-altering side effects from their COVID-19 vaccine. We build bridges between patients and research institutions in order to develop a better understanding of our vaccine complications. We have tonight with us Asher Milgrom. Thank you again for being here. Asher he went to the University California, San Diego with biological sciences, and you got a president's grant for independent research in marine biology. And then you worked with the Scripps Institute of Oceanography, and he started a medical program, the MD PhD, and was on a full scholarship in the neurobiochemistry. But toward the end of his training, Mr. Milgrom here had a near-death experience, and after he developed a rare brain infection. And so I read, after touching the face of God and living, you left the practice of medicine for an entrepreneurial life, developing the science of medicine, which is pretty cool. You went on to the University of Chicago and got your PhD in biomedical sciences. Asher continues to expand his expertise through training in advanced alternative fields, including laser medicine, metabolic medicine, naturopathy, integrative and regenerative medicine. And Asher is now AMA's CEO and chief scientist. He's done numerous talks and appearances live and virtually, and I'm absolutely thrilled to have him here. And on that, I'm going to hand the floor over to you, Asher. Thank you for being here.
Thanks. I am honored, privileged, and James will be standing by in case we have more technical issues along the way. I always start these conversations essentially with a almost like a prayer, that somehow the content and the intent and the spirit of the work that we do, the research that we do, almost all of it self-funded, it somehow transfers heart to heart to all of you in a way that is both inspiring and uplifting without contention, and even the elements that are seem to the allopathic conventional physician as difficult to comprehend or difficult to accept or recognize as legitimate, that all that drop by the wayside, and all of us with open minds and hearts can engage in this conversation. And all of it, of course, in honor and in glory of God, that is the ultimate physician, the ultimate healer of all afflictions. So, having said that, James, I need to go to share the screen. If you will go ahead and do that for me, and there you go. All right, and of course that's in my way. So how do we move that? Okay, perfect. All right, and I just use this down key here. All right, so today I'm going to talk to you all about the 5 integrative modalities that we use in our practice. So first, let me explain briefly that we are, my wife and I, an integrative and regenerative practice. My wife, though I didn't continue with the MD side of my program, I went into the 4th year and decided to just focus on the science and into the PhD area of medicine. My wife did complete and her MD, and she is the medical director of the practice. There she is right there. She is the best thing that ever happened to me, my wife and best friend. And You see here right now injecting ozone paraspinally for someone who's had multiple spinal surgeries. And the results of ozone injections or prolotherapy are quite amazing, but there she is. So our practice is ultimately integrative, which means, in case you don't know, that we will use anything that has clinical relevance and is safe to use. Doesn't matter the source, and so we integrate, every possible modality and method that we can for the patient's benefit. So just to start, given the environment that we're in is so different than the environment we went in medical school, or when I was in medical school 35 years ago, we were practicing medicine in a free democracy. It's no longer a free democracy. The practice of medicine has become very contentious at best and overridden with all sorts of regulatory draconian forces from above. This is what medicine looks like today. And whenever I give this talk, this is the slide that gets the most laughs. The doctor and the patient, as you can see, are not even in the conversation anymore. And those four gorillas, big pharma, the feds, the lawyers, the insurance companies, are screaming at each other, controlling the entire environment. And poor little, healthcare angel is trapped within the bureaucracy of those gorillas. This isn't the first time this kind of thing has happened in our history, but the bottom line is, the federal government now has the authority to force the acquiescence of U.S. citizens to take experimental, untested medicine, or face unemployment, dishonorable discharge, loss of licensing and credentials, etc. Doctors are forced to lie to their patients, fabricate data, falsify statistical analysis, on and on and on. I don't need to spell it out. You all know the story because of who you are. So I believe I know my audience here. I hope so. The conclusion of all this is too horrible to really even contemplate. And the obliteration of citizens' right, the constitutional right to demand accountability, is horrible. And because of all that, informed consent is simply out the window. And hence we have, but I don't know at this point how many hundreds of thousands of people suffering from vax injuries, and that number is only going to increase. There are a number of organizations that have broken that, broken that cage open and let the angel of medicine fly out. Those are some of them. You are actually supposed to be named up there as one of those organizations, but again, given the technical difficulties, we weren't able to put your name up in there in that slide. Good medicine is an art. It requires the freedom to practice and develop the most clinically effective protocols customized for each patient under exclusive control of doctors, not remote bureaucratic forces that now tell us what to do. So our experience with COVID. Now, the 5 modalities that we use for COVID is also the 5 modalities that we use for long haul COVID, whether vax injured or simply a natural progression of the disease. By the end of the Delta emergence, we had treated over, not 1,700, but actually 2,500 patients with 100% success, meaning none of them died. The vast majority of these patients recovered at home using the FLCCC early treatment protocol. Those who are already beyond the 2nd week of their illness and experiencing more serious symptoms were treated in our clinic if they were local. Although the truth is many fly in from all over the world for these protocols which integrate these 5 modalities. We lost only 2 patients who had already been swallowed up by the machine and admitted for ICU care before their families notified us of their condition. They both died alone in isolation while on ventilators. Again, the evil of that story is unspeakable. The credit for this extraordinary success in the early months of the pandemic really belongs to Dr. Zelenko. May his memory be a blessing. And thereafter to the FLCCC, who became our major source of ongoing guidance and courage, really. And of course, to God. And I quote Isaiah there, and worthy of your consideration. The functional approach is really important to understand. And again, I don't know if you're all physicians on this call, or whether some of you are patients without the medical background. So this talk is hopefully, and is though clinically and scientifically accurate and as precise as possible, written in layman's terms so that everyone can understand it, with the anticipation that I'll make this PowerPoint available to all of you, and you can use them in your practices as you wish. During the first few months of COVID, during which we treated hundreds of patients in our office, we experimented with many modalities and various sequences to determine which are the most effective and necessary. The forthcoming 5 modalities were the winners. And of the 5 modalities, in the vast majority of cases, EBU, which is extracorporeal blood oxygenation, ozonation, and filtration, was discovered to be the single most effective treatment with which to initiate the protocols. And we'll get into the details in a few slides. So here are the 5 modalities. Ozone, and there's a variety of ways to administer ozone. EBU, the extracorporeal blood oxygenation, ozonation, and filtration. The IV therapies, which are redox therapies primarily, sodium ascorbate, not ascorbic acid. Big difference. Sodium ascorbate is a very effective redox compound. Ascorbic acid is not. It is very oxidizing and can be painful. And then other redox therapies in the form of photodynamic therapies with photosensitizing compounds such as methylene blue, and also photodynamic biomodulation is without the photosensitizing, and IV drips such as NAD. Hyperbaric oxygen therapy, which I imagine you're all familiar with, and of course stem cells, and there are various sources of stem cells. Autologous don't work for these treatments. Adipose-derived, they simply don't work. We tried them. Allogenic mesenchymal stem cells, which are omnipotent umbilical cord cells, which are very, very effective, and in fact are now approved under an IND, and I'll talk about that later. V cells, allopathic and autologous pluripotent cells, not available to use in this country, but very effective if you're outside the country and you can find them. Stem cells do work, they're just very expensive, and so we leave them as the 5th modality if the other ones aren't getting our patients to the point where we're all happy. The following slides are a quick snapshot of what the 5 modalities look like. Again, there's insufficient time today for a deep dive into the science, so I'm going to give you an overview of what these 5 modalities are. Modality number 1, ozone. I'm going to speak on that one first, even though we use EBU as the first treatment of choice. And because EBU incorporates ozone, I'm going to speak about ozone first. It's very easy to do. You gravity fill a bag with someone's blood, you infuse it with ozone at a particular ratio, typically 40 gamma for the first treatment, 70 gamma. Gamma is a measure of the concentration of ozone to oxygen that the ozone generators create. You have to make sure the patient has sufficient levels of G6PD in their blood. Red blood cells have no mitochondria, can't handle the oxidative power of ozone, and therefore can cause hemolysis. So you test for G6PD first. Very rare that you find somebody with G6PD deficiency. Of the many, many thousands of patients we've treated with ozone, I think maybe we've had 2 or 3 that were G6PD deficient. Unfortunately, there's no in-office tests for that available in the United States. They're available in Europe, can't get them here. Just another one of those regulatory obstacles that we encounter. But you can check for hemolysis by doing dipstick in urine. And so if you give somebody a small dose of ozone first, then you take some urine and dipstick it, you'll know right away if they're showing hemolysis. If they do, then you send them to a lab for a more thorough analysis. That gentleman there, by the way, is the oldest man who's ever climbed Everest. A very interesting, wonderful man. There's also, instead of a single pass where you fill just one bag of blood, you can do something called a 10 pass, which is that device that you see there, this green device from Germany. The blood fills this container up there, ozonates it, and then pumps it back into the patient. And then you can withdraw another quantity, ozonate it, pump it back into the patient. Each one of those is called a pass. And you can do that multiple times, 10, 15, even 20 passes. In Europe, they often do 15 and 20 passes. We usually limit it to 10 or 12 passes. This device up here simply swirls the blood to keep it in hemodynamic flow so as to limit any kind of coagulation or precipitation of lipids that clog up the apparatus. If you're interested in knowing more about the apparatus, Mary-Lise Urania is the rep for this company, and that's her email address right there. I don't know, I do not want to install, remind me later. Okay, sorry about that. Let me just put it this way. We have millions of dollars worth of equipment in our office, all sorts of lasers, hyperbaric oxygen chambers, cryogen chambers, you name it. We have it all. And if we had to give up every tool in our arsenal except one, the one we would keep is ozone. It's the single most ubiquitously useful, easy to use, and no downside to using it whatsoever, except for the case of G6PD deficiency. It is so effective. Oftentimes, all somebody needs is ozone and nothing else. The other one, modality number 2, the overview here is the EBU that we mentioned. Here you see the blood being extracted from one arm, goes through the device here, which ozonates the blood, and then it goes through this filter here on the side, which is the dialyzer, which filters the blood, and then it is pumped back into the patient, and that simply circulates very much like dialysis, except it's not. It's apheresis, and which is very different than dialysis. And it is so effective, and we don't actually know why. We're trying to figure that out, but sometimes, and I'll show you pictures, the gunk that comes out of people's blood, and you see it, of course, in that venous line right there, is astounding. The stuff that's circulating in people's veins, you know, as a result of COVID, long haul COVID, vaccine-injured COVID. You see it in Lyme disease. You see it in a lot of chronic inflammatory diseases. It's extraordinary. It's pretty grotesque, actually, but the results are amazing. Sometimes, you know, when patients come in with massive brain fog, and all that goes along with it, and you all know what I'm talking about, that brain fog will lift in the middle of the first EBU treatment. And the room is filled with, you know, we have a very large IV arena, and there are many patients in there who are halfway through their treatments or at the end of their protocols, and they're all watching the new patient for that light to go on. And when it happens, it doesn't happen every time, there's a cheer that goes on in the room because they all know what that new patient is experiencing. It's quite exciting. Let's see. James, it won't advance. Oh, there we go. IV therapies, sodium ascorbate, as I mentioned, a very, very important redox therapy, specifically targeted at mitochondria, and we'll talk about the need for that. The electron transport chain is damaged by the spike protein. We'll get into that later. And so sodium ascorbate is remarkably effective on that. Again, other IV therapies such as methylene blue also target very effectively mitochondria. It was amazing when I gave this talk to the FLCCC, even though the room was filled with, I'm told, 2,000 physicians, critical care physicians, mainly ER physicians. They had no idea what methylene blue was, even though every single ER has it for the case of cyanide poisoning and CO poisoning and other things like that. It simply delivers electrons to the electron transport chain and bypasses where the chain is damaged so that mitochondria can generate ATP and the patient can recover. As you can tell, I'm really moving as fast as I can here. The other modality that we use also, photodynamic therapy with either blue or red light. And here you see methylene blue is a photosensitizing compound, as well as being used by itself. It absorbs specific frequencies of light and causes the extra amplified release of singlet oxygen atoms that are readily engaged in surrounding tissue. It will kill pathogenic microbes instantly. It kills malignant cells, so it's also a very effective use in oncology. Integrative doctors use it a lot, integrative oncologists that is, and will donate electrons to the mitochondrial ETC, electron transport chain, increase ATP production and tissue repair. And here you can see it being used on someone who's had back surgery. The transdermal penetration of laser light enhances recovery post-surgery of the tissues that have undergone surgery. Here we go. And here's another example of that wristband. The person, as you can see from the IV, is receiving methylene blue, and then they're receiving at the same time the light, which activates the methylene blue as it's flowing through the vascular system. For oncology, there's an incubation time. You need to let the methylene blue incubate for a while before you apply the light. But again, that's a whole 5-hour conversation in its own. Here's another device that does similar, application of the correct frequencies of light. And these pads here can give more than just the one frequency you see. This person, again, we're treating them for kidney disorders. Remarkably effective, and there's no downside to using it. So if you're not familiar with light therapies and photodynamic therapies and photobiomodulation, which again are very similar, definitely worth learning about. This is a Weber laser. Okay, it's different in that it applies the laser energy intravenously. Okay, these little canisters you see up here and all those fibers, each one of them is a different frequency of laser light. That's what the device looks like. And at the end, there will be a little tiny fiber that you insert through a catheter. So a little tiny tip of the fiber optic cable is accessible or exposed to the venous blood flowing by it. And because of that, you can use different frequencies than you can use in intradermal penetration, right? If the wavelength is too short, it won't penetrate the dermis. And so the only way you can get short frequencies of laser energy into the bloodstream itself is with an intradermal application. The Weber device, expensive, well worth it, amazing results. We should have some pictures. Okay, those are the different canisters, each one of them delivering a different frequency of energy. And here you go. You can see there is the fiber optic, and there's the catheter, and you can see the tip of the fiber optic cable. It comes out a little tiny bit from the catheter and is exposed to the venous flow. And here's again a green frequency. Other UV frequencies you won't be able to see because your eye simply can't see it, but there are many frequencies on that device, as you saw. NAD, again, we all know about the Krebs cycle. Again, for those of you who aren't medically educated, again, I don't know exactly who my audience is here. There are many animations available online. I'll show you a couple. Where you can learn about the TCA cycle or the Krebs cycle and the electron transport chain, all of that which happens within the mitochondria to generate ATP, and the electrons that are donated, from NAD and FADH. These liberated electrons flow through the electron transport chain, supplying the needed energy to generate ATP, thus administering NAD to IV. To cells with many mitochondria or to damaged cells with high metabolic needs. especially neurological cells, which have more mitochondria by tenfold than any other cells in our body, is an extraordinarily effective way to deal with brain fog, TBIs, concussive damage, on and on and on. NAD is incredibly effective. The FDA is doing all they can right now to make it impossible to get it. So compounding pharmacies are really the way to go. It is remarkably effective. Now, the reason I keep on talking about mitochondria, and I don't know if I'm going to get there in this lecture because it's so long, is because the spike proteins damage mitochondria, specifically the electron transport chain. Hopefully I'll be able to get to that. But when we talk about the hallmark symptom of long haul is the extraordinary fatigue that virtually almost all of these patients have. And that's because of mitochondrial damage. So to focus on mitochondria is exceedingly important. Now let's see. Again, let me get this thing to move. All right. There we go. Hopefully that will do it. Okay. The fourth modality is hyperbaric oxygen therapy. These are rigid. There are two types of chambers. You know, the rigid chambers like this. Some are made out of metal. They're kind of scary. And some are inflatable. By far, these are the ones that the patients are most comfortable with. And why do we use hyperbaric oxygen therapy? The two methods that the spike protein damages our patients' physiologies is one because of mitochondrial damage. The other is because the spike protein engages the endothelium of vasculature and causes massive inflammation. If you have this massive inflammation moving through the vasculature of a patient and getting into various organ systems, it will cause a reduction in perfusion. And very simply, you won't be able to deliver the necessary oxygen to the tissues that are damaged, whichever organ system gets hit, that these tissues need, and you end up getting progressive ischemic damage of the various organs. The only way to deliver oxygen to an ischemically damaged organ that is caused by lack of perfusion, so you simply can't get blood there. So even if the blood carrying capacity is normal, it simply can't get in properly, you know, to those damaged organs. The only way you can get oxygen to those organs is through hyperbaric oxygen therapy, you know, where the pressure in the chamber, you know, causes the oxygen to dissolve in all the fluids of the body so that you can deliver oxygen independent of blood flow. And the results are spectacular and necessary. Now, 1 or 2 treatments is almost never enough. You're talking about 10, 20, 30, and sometimes even 40, especially for those showing neurological damage, the neuropathies, the partial paralysis. You know, again, the neurological symptoms of long haul are you know, there's just so many of them. And the hyperbaric oxygen treatments are very, very effective. I'll just read that last paragraph. This is critically important in patients suffering from mitochondrial dysfunction, chronic systemic inflammation, and abnormal blood clotting. Don't forget that in addition to the inflammation at the endothelial level, the spike protein causes all sorts of abnormal blood clotting, which adds to the perfusion issue of simply being able to flow blood properly. Well, you know, it's obvious. The functional hypoxia, including ischemic damage to virtually every organ system in our bodies. Come on, I saw it move. It's thinking, it's thinking. James, help.
That I can't help.
Oh, yeah. Okay. I wish I could take questions when this was going on. It is stuck. Okay, we'll give it a second. Let me see what else I can mention here. that is applicable to where we are in this talk? Well, does anyone have a question? You can unmute yourself if possible and shoot a question my way. That is, pertinent to, let's see, it looks like it's working again. Yeah, there we go. Okay, so.
Sorry, I was just also going to say, if you put it in slideshow mode, then you can just hit an arrow key to go to each slide.
Yeah.
Like,
okay, so you want to try that?
Like there's a little screen down at the bottom. If you click that and make it, or you can, there's a bunch of different ways you can do it, but your tech person should probably know how to just put you in presentation mode.
Yeah, I wish I knew.
There we go. Presentation view.
So see slideshow at the top.
Yeah, okay, here we go. All right.
Yeah.
So here again, another view of our chambers. We have 3 of them you saw in that room, so we control them by that control panel over there on the left side. Each of the chambers, you can control the rate of inflation or pressurization, how fast it's pressurized and how far you can go. This kind of hyperbaric chamber is by far the most effective and superior technology available on the planet, but they are expensive. And fortunately, through a foundation, we were able to afford some of these. Now, again, I shouldn't spend much time on this. Usually, hyperbaric chambers are filled with pressurized oxygen. There is no therapeutic benefit to doing that. If you can fill the chambers with hyper clean air, compressed air, and then have them through a mask breathe the pure oxygen, you get the same therapeutic benefit that you would with filling the chamber with pure oxygen without all the dangers. And there are a lot of dangers with compressed oxygen. It's basically, rocket fuel, and it's very expensive. And so what you're looking at here is a proprietary arrangement of compressors and filters and all sorts of things to fill our chambers in the way that provides the safest possible and therapeutic efficacy as is possible. All right, the 5th are again the stem cells. There you're looking at a single cc of not embryonic, sorry, we don't use embryonic, we use umbilical cord and mesenchymal stem cells. We use them a lot. We do a lot of research with them. Vidi Labs is the lab we use. Now, you need to know that I've worked with 6 of the largest labs in the United States, and I was really dissatisfied with their science and with their willingness to do the assays that I needed to know what was going on with those cells and the contents of the exosomes they were producing. Vidi Labs science is, in my opinion, hands down the most advanced. Philip Vidi, the chief science officer of Vidi Labs, is remarkable. I sent him a question, a scientific question, and he'll respond within a week, sometimes a couple days. And the work that we have done together with them over the last 3 years in collaborative research and development has now resulted in the FDA has now approved these particular mesenchymal stem cells for an IND study for COVID-related illnesses. The only one in the world that's received that kind of thumbs up from the FDA, which is remarkable because they make everything else so difficult. It was a miracle. And now they have indicated that they are approving also additional INDs from the research we've done with Vidi for autism and muscular dystrophy. So some good things do come from the FDA, and not many, but some. So this is exciting, and some of these studies will be multiple site studies. So if you're interested, please contact me. All right, the stem cells in general, regardless of the condition being treated, whether neurodegenerative diseases or chronic inflammatory diseases, such as COVID or Lyme or lupus, successful stem cell infusions depend on 3 principal elements: the quality of the cells, obviously, cleansing, detoxing the physiology that would impede or damage the cells themselves, especially heavy metals, chemotherapy, pesticides, etc. You got to clean out the patient before you spend the money on the cells. Chelation therapies generally will do it, as well as ozone and EBU. Preparing the physiology so as to have a happy, nourishing, and stimulating environment to receive the cells and promote maximal regenerative outcomes. There's a lot more to discuss here. We simply can't do it in a public forum because of FDA regulations that actually don't even allow me to use the term stem cells, but because I have an IND backing me up, I can. It's craziness out there. Stem cells are alive and they're responsive to spiritual energy. So what you see here is a patient holding onto the vials and as they thaw, and praying over them, blessing them. These are parents of a muscular dystrophy child. By the time these families get to us, they're broke and desperate. And it's a beautiful thing to watch. The infusion is very simple. A simple push will do it. And you can follow it with ozone, with, again, sodium ascorbate and high potency vitamins and other things that nourish those cells and encourage their regenerative activities. They are highly anti-inflammatory. So people come in with, for instance, massive lupus, and again, that's a different conversation how we treat lupus with stem cells. The results are simply spectacular. The inflammation from lupus will reduce to half with a single treatment of stem cells, and then you do 2 or 3 more, and many of the patients never have to come back. We cannot explain it, but the data is, patient records are empirical. So I don't know what else to say. Stem cells are quite magical. Now, a major cause of all the symptoms we see in COVID patients, again, are from dysfunctional mitochondria and systemic inflammation. Both are caused by the COVID spike proteins, whether contracted by the virus itself or by the vax. These spike proteins are everywhere, and therefore the target of all 5 of the modalities that I just gave you an overview of are mitochondria and systemic inflammation. Now, from teeny boppers to members of Congress, everybody is dying to discover the answer of 2 life-transforming questions. Number 1, is Elvis really dead? You're welcome to laugh. And also, what are the detailed protocols that cure COVID? This is why you're all on this call. Fortunately, there's a simple and elementary answer to the COVID protocol questions. There are none. There is no simple protocol that works. Let me explain. As yet, there are no labs that definitively and accurately identify, qualify, or quantify the presence and the extent of symptoms associated with COVID, long-haul COVID, or vaccine-injured COVID. As we all know, the vast majority of those who test positive have no symptoms at all, while others may have debilitating symptoms associated with a specific or singular organ system that got hit with the spike proteins, or on the other hand, others may be hanging by a thread due to the progressive dysfunction of multiple organ systems that have been hit, as there are no labs to tell us what to do. Now, the allopaths on this call, or the allopaths at the FLCCC, hate this because they often don't treat the patient, they're treating the labs. We don't do that in integrative medicine. We treat the patient. We look at the labs. If there was some labs that are important to give us some context of what's going on, but in COVID, there are no labs, that really give us a picture of what's going on. So the only way to assess and ultimately care for a COVID patient is by utilizing a functional approach to develop a customized real-time protocol for each patient as a specific and unique individual. This is old school medicine. Note, regarding the labs, there are none that can penetrate the utterly confusing and unfathomably complex Gordian knot of symptoms. How many times have your patients come to you after they've already been to a dozen doctors who've done, I don't know how many dozens of labs, and they say all the labs came back normal, and yet they walk into your office and they are wrecked. Even Dr. Bruce Patterson's work on the immune dysregulation caused by the spike protein, which is brilliant work, that gives us some degree of confidence that the patient is in fact experiencing a long COVID syndrome. The labs that they do are very expensive, however, and kind of hard to get in line for them. The labs give us little guidance of how to untangle the Gordian knot. So, okay, now you know that they're having COVID long haul, but you knew that already. So I'm not sure the lab helps you, and it just costs time and money. Here are the 200 documented symptoms of COVID and long haul and vax injury. That's just one page of it. Here's another page of it. There are so many. So systemic inflammation plus mitochondrial dysfunction plus abnormal clotting plus dysregulation of the immune system, all of which can hit any and all organ systems, each of them to different degrees, creates an unmappable jungle of over 200 symptoms that no single protocol can possibly address. Therefore, a functional approach to assessing the COVID patient upon their initial consultation and determining which of the 5 modalities is likely to address the most severe of their symptoms as your initial clinical concern requires a functional analysis. I can tell you we tried so many different things in the beginning until we got a hang of it. And a lot of it was intuitive. We may have furthermore, ongoing assessments during each treatment and subsequent to each treatment during daily follow-ups. I hate the workload, but it's necessary, is the only way to assess what is actually working or not working. We have made no clue and are often surprised by which of the modalities ends up being the most effective at a particular point in the patient's recovery. So again, the EBU has proven to be definitely the most effective as the first of the modalities. So every patient that walks in, we start them with the EBU. We filter the blood, get the gunk out, and I'll show you the gunk in a minute. Once again, there are no labs to tell us what to do. The only way to assess, ultimately to care for COVID patients, is utilizing a functional approach to develop a customized real-time protocol for that specific individual. All right, now,
Asher, can I interrupt you 1 second, please? This on your screen, on our screen, we see give subtitles a try on the bottom left-hand corner of the screen. I don't know if you're able to get rid of that.
I don't see it. Yeah, maybe
yeah.
Gives a try, and I have no idea what
it says give subtitles a try. So in the middle of your
oh, there it is. I see it.
If that can somehow go away.
Well,
let's see. There.
Okay.
Okay.
At least we can see it now.
Okay.
Perfect.
Great.
Sorry, thank you.
Right, we're good. So I have no idea what that was.
that was Microsoft doing its thing.
Oh, that was Microsoft. So all of you Apple devotees, I'm gonna have to convert to Apple. You heard it. Yep. They heard it. Mea culpa. I don't know what else to do. All right. My kids will be very happy. All right. So you're the first to hear the next 2 slides. Breaking news flash number 1. AMA Regenerative Medicine has undertaken a new project to develop an interactive database which will track patient systems as they undergo treatments. The key metrics are including the following: patient profile, obviously, list of symptoms that the patient is experiencing. So out of those 200, just the ones that are experiencing are the ones that are activated in this particular patient's interface with the database. The daily scoring of symptoms. They have to score their symptoms every day. So from a 1 to 10, where are they with whatever symptom? And they get an email every day or a text every day reminding them to do it. Okay, and then of course, ongoing administration of the protocols. All that's tracked. The database is designed to be queried in order to reveal the patterns of what works and doesn't work, in order to help physicians discern and customize the most efficacious protocol for each individual patient. Now, when I presented this to the inner circle of leading scientists at the FLCCC, they were just jubilant with excitement because all of them are simply overwhelmed with trying to figure out what the heck is going on. You know, what is working and what is not working. And this database hopefully will reveal the patterns so we begin to see, we begin to be able to discern, you know, with patients when they come in, given the particular grouping of symptoms they have, and which ones are the worst, and in order of priority, we'll know from thousands of patients using this. This will be open source. You will all be able to use it. So you'll be able to log on, assign a patient, access, the patient, and you will have the only ones that will have access to their page. And you will be able to begin collecting this data, query the data. We'll find some way for all of us to coordinate with the FLCCC so that we can do a meta-analysis of what's going on. We're very excited about this. I don't know why it took me so long to think of it, but we're doing it. And we're very excited about it. We're hoping to have something literally in the next 2 to 3 weeks, at least the initial prototype of how this will work. And that's breaking news flash number 1. Breaking news flash number 2, metabolic medicine. Okay, as previously stated, currently there are no labs that tell us what to do. This is true. However, mitochondrial function is not only the critical target and the source of many of the COVID symptoms, it is also the foundational principle of health in general, well-being, and longevity. If we could track mitochondrial function with empirically derived objective metrics, we could thereby know how a patient's health is in fact empirically, okay, not subjectively, but objectively responding in response to the protocols. Patterns revealed by the database and the data generated by empirically derived mitochondrial function should match each other. Now, Dr. Schallenberger of the AOT, I hope you all know who he is. He's kind of like the grandfather, or what's the other term I use? Godfather of ozone in the United States. He has developed a proprietary apparatus for measuring mitochondrial function to achieve exactly the above. Empirically derived objective numbers that tell us precisely how a patient's mitochondria are functioning. And after each treatment, as we're tracking their system, their symptoms, well, we should be able to then have them come in, do one of these tests, okay, which includes a VO2 and a bunch of other things, and track their mitochondrial function along with the database, and thereby give the allopathic world something to chew on. I'm very excited about that. And I'm very excited about the fact that it will help us customize the treatments, both functionally, our functional observations and the patient's observations, as well as some kind of empirical, objective numbers. The synergy between real-time functional analysis provided by the database and real-time empirical laboratory measurements of mitochondrial function represents the holy grail of functional medicine. And there may be some of you functional physicians on this call. I hope you're as excited about this as I am. All right, number 3, the great news about these 5 modalities, beyond the massive clinical experience that proves their effectiveness, is that we know that none of them can hurt your patients. They are not only safe, but also promote health and vitality for your patients, whether they're healthy or sick. So given number 3 above, the physician for which all this is unfamiliar, you can breathe a deep sigh of relief. These modalities are fantastically healthy for every one of your patients that walks through the door, providing extended vitality, longevity for your patients, and righteous income for you, their physician, okay? And you can do it with impunity. You can begin experimenting with these things because they are so safe, okay? Above, all of the above foundational principles apply primarily to patients who are not recently infected with COVID. Those who are recently infected with COVID, again, that's not your issue because you all are vax injured, but again, the recently affected may show up through your doors. These modalities are healthy for everyone. They require multiple office visits. They're typically not covered by insurance. On the other hand, patients who only recently contracted the infections can be readily and effectively treated at home by using the FLCC protocols for early treatment, which are, again, remarkably effective, using primarily or almost exclusively repurposed medications. And again, the FDA is forbidding the use of a whole bunch of them. Just do it anyhow. That's all I can say. All right, we believe we have experienced that successful clinical outcomes start long before treatments being established in the patient. Okay, well, let me backtrack. First, this is about consultations and informed consent. The healing of the patient starts at the level of the consultation with an alignment of mind and heart. I don't want to go through this right now because you can read it on your own later, but I spend an hour with every patient who comes through the door discussing what's going on with them. There's no charge for that. The alignment of mind and heart is exceedingly important for their physiology to be on board with any protocols that we do up down the road. I really believe that alignment is necessary before the body will participate. And so we take I use lots of slides from this presentation to explain to them what's going on and why the protocols work. The patients walk into the clinic deathly ill and feeling that their body and the divine intelligence that created is failing them. Our goal, even before they receive treatment, is to engage our patients in the mystery and encourage them in recognizing the miracle and the unfathomable intelligence that created them, because it's going to be that same intelligence that will heal. Again, the major cause is dysfunctional mitochondria and systemic inflammation. Therefore, the target of all 5 modalities is the mitochondria and systemic inflammation. Now, at this point, it's back to school. Now, what time? How many more? It's already 12. Let's see. We're already at 45 minutes into the treatment. What time is it?
55.
We only have 5 more minutes. So you want to just go to questions. Let me just, I'll just screen through these things very quickly, because even at the level of the most prominent allopathic critical care doctors, going back to school on this was really important. So many of us have forgotten how mitochondria functions. And in the last 35 years, there's a lot more about it that we know now that we didn't then. So I take people through a back to school. I needed it myself to remind myself how and to learn at a deeper level how mitochondria function, how the electron transport functions. And again, it's as Einstein said, if you can't explain it simply, you don't really know it. Very important that I can explain it simply to my patients. And again, the patient alignment of mind and heart. You know, the miracle of the body with its 50 trillion cells. Every cell in our bodies, again, these are numbers you may not know, does 20,000 biochemical reactions per second. Most of our patients think I meant per hour. No, every cell, 20,000 per second. And very important for me to get them out of thinking about their misery and to thinking about the miracle that is their body. And again, the mitochondria, about ATP production, where that occurs, how it occurs, where the electron transport functions. What's important here to understand, again, is that the electron transport chain is composed of a set of cytochrome protein complexes 1 through 4. They are all of the heme family. The reason this is so important is the spike protein is, among its other toxic effects, is also a prion. And it specifically engages and distorts heme molecules. So it gets into the mitochondria, it's tiny, it penetrates right into the mitochondria and messes up the electron transport chain. Okay, so you have to deal with that. And that's why so many of the things that we do, you know, are protocols and modalities that will deliver electrons directly to the electron transport chain, bypassing where it's damaged, right to cytochrome 4 and ATP synthase, so that the mitochondria that's damaged can in fact still generate ATP. NAD does that, ozone does that, the photobiomodulation does that. That's why oftentimes patients will wake up and the brain fog will lift and the energy will return even after one treatment. It's never enough one treatment, but you see the changes that quickly. So again, the Krebs cycle and and where the electrons come from to the electron transport chain, how they're delivered to the electron transport chain, and then of course the electron transport chain right there. Worth reviewing. I had a great time reviewing it. I was stunned by how much I didn't know, and humbled by it and excited by it. This is an animation, which I will not play now, but again, I'm going to send this to you. You can play this animation. Oh, it's playing by itself, but you can show this to your patients so they understand, again, how the electron transport chain works. It is, it's miraculous. James, I'm stuck again. There we go. Oh, there we go. There's another animation that I just passed, which is from Harvard, which gives much more detail about it all and is animated and voiceover, which is remarkable. So we are at the end of the hour.
I would do that.
What's that?
I would do that.
Okay, so let me, again, spike protein. Spike proteins are released every time the virus penetrates the cell. There are certain immune cells, this is Dr. Patterson's work. Again, we're reviewing his work. These cells are gobbled up by certain class of immune cells and delivered throughout the body. The question, of course, is why does the spike protein persist in our bodies? One of the reasons is that the immune cells that gobble them up don't recognize them as natural. These are synthetically derived molecules. They were engineered, there's no question about it. So normally the cells that gobble them up, I'm forgetting the name of that cell right now, they will dissolve anything they gobble up and disappear in a couple of weeks. No, it's not a macrophage. Let me see. Oh, here we go. This is the immune cell that I'm talking about. I've written about it someplace else. These cells normally only live for a couple of weeks, but once they've gobbled up the spike protein, the spike protein is not dissolved in their internally. It stays active and causes massive inflammation of that cell. That cell then patrols the body, delivers inflammation to organ systems all over, for 3 to 4 months. That's one of the reasons that long haul COVID even exists, because the spike protein persists long after the virus is gone. We don't really know. Now, the question, of course, becomes the following. So they come in for a long haul treatment and we treat them. And within 2 to 3 weeks, they feel like they're alive again. They are 90% to 100% recovered, the vast majority of them. Given that the spike protein has this tendency to stick around for a long time, will the long haul return? Are they going to come back? All I can tell you is that we've treated hundreds so far. None of them have had to come back. We don't know why, but we know the results. Now, they may get COVID again, especially given that we know that people who are vaccinated are more susceptible to the ongoing progressive variants of COVID. They may get COVID all over again, but if they catch it early enough, it won't develop into long haul. But this is, again, a very effective animation, for our patients. Monocytes, those are the cells that normally gobble up protein debris that's floating around the vascular system that's not supposed to be there. Again, the spike protein is also a prion. I mentioned that. Number 4, spike proteins bind to blood coagulation factor fibrinogen, induces structurally abnormal blood clots with heightened pro-inflammatory activity, disrupting blood flow perfusion to organs throughout the body. and hence the EBU filtration is so important. I'm going to go straight there. How ozone works. This is the section on that. By the way, if you look at do a live blood analysis of your long haul patients, you'll see all these roulet formations. Their red blood cells are suffering. Their ability to carry oxygen is highly depleted. A single dose of ozone will fix that. After ozone and before ozone. Let's see. Again, all the things that ozone does are all listed there. Again, I'm trying to be conservative of your time. I just want to show you all the ways ozone can be administered. Again, we don't have time to go into it, but I do want to show you, again, systemic inflammation. Okay, that dialyzer, that filter. This is all about ozone. Again, that's the swirling that I mentioned to keep the blood and hemodynamic flow, very important. Okay, EBU, there it is. Rarely have to go beyond 5 treatments. The gunk that comes out of people's veins is extraordinary. There it is. Now, you've ever seen that come out of a normal patient's vein? Never seen anything like it. It's both disgusting and exhilarating all at the same time to see it come out. There it is again. And some of these, okay, these, you can see the gunk just, I don't know how people can function with that stuff in their vasculature. Manage to circulate throughout their entire body and somehow not cause, CVAs. I don't know how it does it, how our body manages to deal with it, but that typically comes out of our long haul and COVID patients. Wow. Okay, it is amazing.
Are you seeing that in your vaccine injured too?
Absolutely.
The gunk, I'm sure.
Absolutely. Look, the spike protein doesn't matter where it came from.
Yeah.
If it came from the vaccine or the virus, doesn't matter. It's causing the same damage, exactly the same damage. And then after the booster, it's even worse.
Mm-hmm.
So,
Dr. Richard Chen is asking, by what mechanism do you think that the EBU is helping?
The filtration. Okay, you see that filter right there? That filter is the aphoretic filter. Okay, so let me show you some pictures of it. Okay, there it is before the blood is flowing. There it is during the treatment. You can see already that it's turning colors. Mm-hmm. Okay, it's becoming white. That white junk in there is the gunk that's coming out of people's veins. Okay, this is what we found. We sent it off to analysis. Unfortunately, very expensive, $5,000 to analyze, one of these filters. The lab opened it up and analyzed the contents. Lots of biofilm. Now we know biofilm happens with various infections like Lyme. Now we know that biofilm also is inherent in COVID infections. Again, I don't know why, but the body is filled with biofilm. Okay, microclots, we know that. We see them in the filters. Those abnormal fibrinogen clots, we see those in the filters. Opportunistic bacterial infections, we see those in the filters. And extracellular traps, which I did not expect. Okay, now also microplastics and heavy metals are also trapped by the filters. And here you can see it. So here are some of the analysis under different conditions. This is, I forgot which stain they're using here, but here's the erthidium bromide stain. And you can see again what the lab found. The extracellular traps, again, you're looking at it. You're looking at that huge masses of fibrinogenic abnormal clots. Here again, those are the strands, the filaments in the filter itself. Again, more of the same. It's been a long time since I've spent time in the lab, so I'm glad others analyzed that and it wasn't dependent on me analyzing it. Again, there's more and more. This is the same patient's blood after an EBU. Perfectly normal, clean blood with perfectly normal red blood cells without Roubaix formations.
How many treatments was that?
One, just a single treatment. Okay, now they need more than one. They need somewhere between 3 and 5. So after the 3rd treatment, if we're not seeing any more gunk come out of their blood, then we stop because the EBU is expensive, the filters are expensive, and we just continue with ozone, which is much less expensive. So, I mean, managing the patient's expectations, managing their protocols, managing their physiology, and managing their wallet is very important here.
Yeah.
Okay, now patients will pay anything to get rid of this stuff, but I don't want to take advantage of that. So if after the third, as soon as the gunk starts coming out, we'll just, again, the EBU does ozone and filtration, so we'll go to that 10-pass device, which is just ozone without the filtration. And here's another patient. You'll see the same thing with every single one of the patients, the same thing over and over and over again. And there's their blood at the end of a single treatment. So again, look at that. I mean, it's like a snake coming out of their veins. I mean, the first time we saw it, we didn't even know what to say. But we see it with every single one of our patients, and we've done thousands. Hundreds of COVID and vax injured, but thousands of patients, including the Lyme patients, and never seen anything like this. Never seen anything like this.
I'm going to throw another question at you.
Please, please.
Okay, so Dr. Ann Kelly is asking, while if spike protein impairs the electron transport chain, perhaps hydrogen water and D-ribose might be beneficial as they bypass Krebs cycle and help ECT.
Indeed, both of those are safe to use. So I would try it. There's only so many things we can do at once. So, but that is something worth experimenting with. And even hydrogen peroxide. Now, there are some people that have such poor vein access that you can't get a large enough catheter anywhere in them to draw blood out, either for ozone treatment, or for an EBU treatment. It's just, you just can't get access. So if you hang a bag of ozonated saline or saline with hydrogen peroxide, all you need is a tiny little, baby needle, to get both of those or either one of those compounds in their blood, which will work, again, directly on supporting the electron transport chain. Anything that is a redox, redox compound that can penetrate the mitochondria, not all of them can. Peroxides, small peroxides can. By the way, that's one of the ways that ozone works. Ozone interacts with lipids in the blood and creates little tiny peroxides, which have a long shelf life. They circulate for a good couple of weeks. So the effects of an ozone treatment isn't just in the office. It will continue to help the patient for a couple of weeks. Though, generally speaking, we try to accelerate this. If someone is really sick, we'll do 2 EBUs per week. Try to get them over the hump as quickly as possible so that at least they're functional. I'm sure you've seen it, patients who come in a wheelchair. They are so debilitated from this, they can't even stand to pee, for men. And they're bound to wheelchairs. But within a week or two, they're out of the wheelchair. If you go to our website, where are the videos on the patients? The
AMA Regen Med.
So if you go to our website, it's in the beginning slide, AMA Regen Med, and where do they find, what tab do they find the?
Under COVID.
So under COVID, you'll find patient testimonials
Patient recovery.
and patient recoveries, and they'll tell you their story. We don't edit it. It's raw. They'll tell you exactly how they were when they came in and where they are after X amount of treatments. We hide nothing. We want this to be as transparent as possible. We had a lot more of these videos up last year, and the FDA came at us. They came at us shooting for bear, loaded for bear, and we had to take a lot of them down. And then,
Someone's not muted.
yeah, and cover up some of the words, and because we were using words that they forbade us to use, but the results are simply spectacular, and you'll see the same.
Couple more questions because we're
Yeah.
running late on time here. So you had mentioned, well, I lost it. You mentioned can you mention which article you referenced that deals with how spike protein affects mitochondria and ECT?
Oh boy, you know what? I'll send you a list because it's already
Okay.
way back in the. I'm afraid to go backwards.
Right? Okay, cool.
By the way,
So
here's the analysis, by the way, of heavy metals. So every one of these, aluminum, arsenic, cadmium, lead, and mercury, all significantly reduce with the filtration. So up until now, those who deal with heavy metals, chelation has been our only tool. And the now EBU, apheresis, has been proven to be very, very effective, on heavy metal clearance. It's remarkable, 140 down to 9.7.
do you know, Asher, do you know of other EBU centers around the country? They're specifically asking about Texas and in New York City.
Yes, the person to call, again, her name is Angie Valdivieso. It was in that initial slide way back.
Okay.
And the name of her company is detoxforlife.com. Please reach out to her.
Okay.
She has done a remarkable thing. She is the one who imports these devices from Malaysia. And the truth of the matter of the quality of the device from Malaysia is that the quality of their engineering and the quality of their manufacturing and assembly was poor. Fortunately, the dialyzer, okay, there it is right there. The dialyzer, the filter is made in Japan. And we all know that Japan's quality control is as good as it gets. But the other part of the device, the motor inside that moves the blood and everything else, poorly designed. So one of mine, I have 3 of them. So, and they're working nonstop. One of the devices broke down. I opened up the cover. I looked inside. I said, "Oh my God, there's nothing to this device." I called her up. I said, "Angie, a good engineer could redesign this device, for far greater quality in an afternoon." And so happens that both her sons are engineers, and she did it. And what you're looking at right here is the new device, okay, that is manufactured here in the United States by Angie's team. She created a company that's now manufacturing them. And again, the dialyzers still come from Japan. And you call her, and she not only sends you the device, but she comes out herself and trains you on the device, trains your nurses on the device. She just hired a physician who was so excited by this that he decided to sign on as to help her do that, to show up at a doctor's office and train the staff on how to use it. It's not difficult. And by the end of the day, you'll be treating patients with this device. So please call Angie. She's a delight to work with. She's an RN with cojones,
Beautiful.
It really.
We love those RNs with cojones.
Absolutely. I remember when I was, again, still in medical school on the floors, where did I learn my clinical protocols and my clinical etiquette and everything else? It wasn't from my attendings. It was from the nurses.
Yep.
I knew which nurse to go to.
That's right. That's awesome.
Yeah.
There's one more quick question here. I don't know if this is the proper forum for it. She's asking about the complication of the MTHFR. So I don't know if this is something to discuss here.
We have a lot of people looking at that. It doesn't affect negatively any of these protocols.
Okay.
We've treated multiple patients with MTHFR issues. The protocols still work, but as you know, they have other issues.
Right.
So it may take longer to get them through.
Okay.
What I can tell you is that my wife and I, Dr. Pian and I, decided halfway through this that we can't let people's financial challenges get in the way of healing them. So we dumped half of our life savings into a foundation. We asked the patients who are well healed to help. We turn no one away. And those MPFR patients need more help.
Yeah. So thank you so much, Asher. I just can't thank you enough for, number one, agreeing to do this at such a short notice, but just being wonderful. And as you said, you know, we're doing God's work, but you're doing God's work, and you don't take advantage of people, and it shows, and truly appreciate everything you're doing. With that, I'm gonna wrap up for the evening. You're gonna send me the slides if you haven't already, and I'll make
Right.
sure they get out to those who would like them.
Right.
And I just, again, I thank you so much. So I'm gonna wrap this up. And I appreciate everyone's questions. I'm sorry if I can get you in contact or, you know, somehow do this individually, then we're gonna do this, but
I'm available for future, you know, discussions,
Fabulous.
conferences, whatever you need. And I'd love to be included in your other conferences to listen in and learn.
Yep, you're on the list now. You're on the
Right.
list for sure. So absolutely, you'll get future emails for that. So our next call is going to be on the 24th of January. And it's going to be Keith Berkowitz from New York. He's in Manhattan. He's an integrative functional medicine guy. He's going to be talking about low-dose naltrexone. So that's kind of his specialty. And he presented that at the FLCCC conference also. So Keith is a great guy, and he's going to present that for us next call, January 24th, 7 p.m. Eastern time. So with that, I'm going to say thank you so much again, and thank you to everyone who was on the call tonight, and I appreciate your time. So
It's my honor. Thank you.
okay, all right. God bless. Good night.
Bye.